but it doesn't work, Lily. Not sure why. And then I just present from here. Is that correct? Okay, not sure why it didn't work. So, um, anyways, thanks for the moral support. Thanks, Lily. No, we can still do it because we're presenting at the digital pedagogies thing out in Scarborough, so it's a local thingy, and we can still do it. Everyone's schedule seems to be crazy though, so sorry. In 1 minute, yes. Yeah, yeah, yeah. It's still possible. Yeah. Sure, we it may not be neurosurgery. Doesn't have to be the brain. It could be the spinal cord, for example. Yeah. Yep. Yep, I think so. Probably wait a while because there's going to be a lot of emails regarding the upcoming term tests and stuff. So yeah, please. Sorry. Any chance you subscribe to any of the YouTube channels that we have for the things? Like, have you looked at any before? Can you see if you can hear this online right now? Like, if, yeah, it should be. If it's working this time, it should work. You should be able to see the new one. Sorry? No, it's the physiology one. U of T physiology. Yeah. Yep. Yep. Can you see it moving? Okay, hi everyone. If you don't mind, I'm going to try to get started a little bit early. Even though we don't have a lot of slides, there are actually a couple of complex slides that I need to spend a little bit more time going through than we normally do. I was told by your classmate that today's YouTube feed seems to be working, so there should be a YouTube recording going up online through the U of T Physiology YouTube channel. So hopefully that'll work all the way through to the very end. So apologies that the last recording didn't seem to really make it online. So I'm going to also ask you if you have the opportunity on Monday, we'll be having an extra help session. It'll be the same format as before. I'll take you through a couple of slides. It'll be in Wetmore Hall in room 54G. And if you have any questions, to show up at about 2 o'clock, and we'll go for about an hour. It will also be streamed live on one of these channels that I've had before. I'm going to try to do it a little bit differently for you for the term test as well as the final exam. And I will give you some practice questions from previous tests and from previous exams that you can try and work your way through. I'm going to ask you not to share it on any paid site. So if you have like a student buddy account or you're doing it through something like that, please don't. It's for your study purposes. And if it's something that every year students are finding useful, we'll continue to do that. But I also don't want any paid sites to benefit from something that a student has uploaded. And I do check those frequently, and I'll be very disappointed, for example, if we see some of the practice questions being uploaded online. So I will make those available. You can ask me for a copy of them. They'll be available as of Thursday morning, and I'll give them as a PDF. You can share them on Facebook with the rest of your classmates. You can work on them independently if you want. Whatever you feel most comfortable with, just email me and I'll send you the PDF package for that. So today we're going to talk about something a little different than what we've been looking at before. And we're going to talk about the autonomic nervous system. And the autonomic nervous system is just like any other type of nervous system that we've been looking at. So it's the same sort of setup. We still look at things that are occurring within the spinal cord. We still have neurons communicating. We still have neurotransmitters being released. And in fact, we can start building autonomic reflexes using this autonomic nervous system, just as we've seen in other parts of the CNS. So I'm going to take you through some really important and key features associated with the autonomic nervous system that make it unique. We'll be looking at the two different branches of the autonomic nervous system, the sympathetic versus the parasympathetic. There are some things that are common. There are some things that are different between the two. You should be very clear on what the commonalities as well as what the differences are between the two different systems. We'll spend a brief time going over the enteric nervous system, how the autonomic nervous system controls the gut nervous system, but we'll just cover that in passing, and then we'll go through one example of an autonomic reflex, and then we'll start looking at a higher order of processing, just like we've done with other systems earlier on. We'll look at the autonomic centers in the brain, just like we did with the motor cortex and others, and then we'll talk about specific aspects of the autonomic centers in the brain that help you, without you realizing it, to maintain homeostasis. And these autonomic centers are built on these autonomic reflexes that are built on the general features of the autonomic nervous system that allow you to maintain homeostasis, just like we've seen with other systems. We have a hierarchy that we can work our way through. So I'm going to give you an overview of the different parts of the autonomic nervous system. Again, simply put, it's the sympathetic versus the parasympathetic. I'll give you some ideas of what the differences and what the commonalities are. So this sympathetic nervous system, as many of you know, is that fight or flight. It's part of this autonomic nervous system, and it's really involved in maintaining your homeostasis. Both systems are involved in maintaining your homeostasis. So it's the housekeeping, and there are different aspects of the housekeeping autonomic nervous system that is important, and we'll cover both aspects of these today. So in some ways, again, it's like a reflex action. You don't have to think about it. There's different nervous system impulses that go to your muscles that as soon as you start exercising, as soon as you go for a jog, because your muscles require more blood supply, the autonomic nervous system is going to, without you thinking about it or with conscious effort, automatically divert blood to the muscles and start to stimulate these sweat glands and everything else that's involved in exercise. So emotion, again, related to how all of these different emotional components will also affect the autonomic nervous system. We'll see that at the end. And again, another place where the autonomic nervous system comes into place is when you are regulating blood pressure, and I'll give you this as an example later on today as a higher center for the autonomic nervous system. Have you guys all seen your midterm exam results? Yes? Were you happy with your midterm exam results? So half yes, half no, and some not sure. That's okay. I want you to do better on your next term test. So I'm going to, as we go along, give you some examples of things that you should be spending a little bit more time on that you'll either see on the upcoming term test on June the 9th or on the following final exam. So this again is just to give you an idea. You should not spend any time memorizing this. You shouldn't memorize that previous slide either. It's really to give you this idea, just like we've seen with the spinal cord and then CPGs, and then from CPGs getting into the motor cortex, that we can do exactly the same thing with the autonomic nervous system. Instead of dealing with the skeletal muscle and instead of looking at sensory receptors, we're now going to be looking at stuff that's happening inside your body, the parts of the gut, the muscle, all of the internal organs that you need to be regulating. And these visceral effectors work in exactly the same way. There are short reflexes that are involved. So if you distend the stomach, what's the normal response of the stomach? It's to start digesting food. There are longer reflexes, and we'll see that there are higher levels of processing. There are areas in the brainstem that are going to be really important, especially areas like the periaqueductal gray. And we'll see that there are areas that are higher up, like the hypothalamus, that are going to be really important within the autonomic nervous system. So it's no different, in other words, this hierarchy that I'm showing you, it's no different than looking at the same sort of organization that we looked at with short reflexes in the spinal cord, higher transcortical reflexes, and then how higher levels of the brain can interact in influencing all of these other responses. So the old view when I was an undergrad was that the autonomic nervous system was the involuntary nervous system. It's not an inaccurate description because you don't have to think about how you're diverting blood to your muscles, and they're skeletal muscles. You don't have to think about how to regulate your blood pressure. It's all done automatically for you. So in many ways, it is involuntary. There is no volitional control of this. And it's the part of the CNS, as I mentioned, that controls all of your internal organs, how your gut works, the endocrine function and how your endocrine glands are going to work, how your blood flow is going to be affected, whether the smooth muscle in your eye is going to dilate or whether it's going to contract, all the viscera, and it maintains the interior milieu, or le milieu intérieur, all of your internal components at a precise level. So your body temperature being part of this as well. These are two efferent systems, whether it's sympathetic or parasympathetic, it's always outflow. So efferent meaning outflow, it's from the central nervous system, whether it's spinal cord, brainstem, or a higher part of the CNS, it's all outflow or efferent from the different parts of the CNS. And the sympathetic is really stress related. When you are stressing out your body, either emotionally stressing your body, physically stressing your body out, mentally stressing your body, it's the sympathetic nervous system that gets turned on. And then it's parasympathetic, it's the restorative, it's the nutritive or housekeeping part of the autonomic nervous system. Both have to come into play. Can you think of a reason why we need both parts? Any ideas? So we have two different outflows, and I'll show you how they kind of overlap with each other, and you'll see that there are many common targets for both systems. But why bother having two different systems interacting with the same organs? Why not just have one system that turns on, and then when you don't need it, turns off? Why bother having two? Can you think of a reason why? Or a simple reason why? So this gets back to what your sympathetic nervous system is really about. So imagine, for example, that this part of your nervous system is always turned on, right? So imagine that you're at an institute of higher learning that is really stressful for you, and you have to be there for 4 years, and you're under stress for 4 whole years. And imagine what the toll that would take on you. It's like exercising 24 hours a day, 365 days a year for 4 years. Is that something that's beneficial to you? In the long run, it's not. And you need this parasympathetic nervous system that controls after you've been stressed out, after you've been exercising, after you've gone out and had this fight or flight response. If you don't have the parasympathetic nervous system kicking in and causing you to eat, rest, and relax the muscles and to replenish all of the glucose, restore your glycogen levels, without that, actually, you would be a mess. And I will show you a specific example of this a little bit later on, where this parasympathetic system involving sleep and other things, if you don't have it, you can literally kill yourself. And in fact, that's exactly what happens. So we'll spend a little bit more time on these different subsystems, and we'll go into the fight or flight or the rest and digest systems really briefly. You'll get this later on in PSL301, where you'll learn about how these parasympathetic inputs into the digestive system help in the digestive process. And we'll just mention this in brief in passing. So the next two slides, this slide here and the next one, are going to be really important. So again, by the end of today's lecture, you're going to know this anyways. You don't have to memorize it. So we are looking at the autonomic nervous system to control your blood flow, your heart rate, how you breathe, whether or not you're digesting food, some of the urinary functions, the reproductive functions. All of these are visceral, they're inside your body, and it's the job of the ANS to control all of these different things. So the overview of both systems is very, very simple. So we have, as part of the autonomic nervous system, whether it is sympathetic or whether it's parasympathetic, it's exactly the same. When we look at the lowest level, that coming out of the spinal cord, we have these preganglionic neurons coming out of the spinal cord, and these preganglionic neurons will send axons out, and they will make synaptic connections into the next group of neurons or the next group of receptors that are part of the postganglionic response. And these preganglionic neurons, whether we are talking about the sympathetic nervous system or whether we're talking about the parasympathetic, all of these preganglionic neurons always release ACH. So at this particular synapse, it's always acetylcholine that is being released, regardless of whether it is sympathetic or whether it's parasympathetic. Is that clear for everyone? So here's a commonality for the system that we're looking at. Okay, so they all originate in the CNS. All the preganglionic neurons release acetylcholine, but what differentiates the sympathetic from the parasympathetic is what happens at the postganglionic level. Okay, is that clear for everyone? There are a couple of other features that are really important in this diagram as well. And again, I don't need you to memorize all of the details. So again, don't memorize all of these target organs, but it's to give you an idea of what these target organs are. And you can see that on this side, we have the sympathetic nervous system, and on this side, we have the parasympathetic nervous system. And you will see that whether we're talking sympathetic or parasympathetic, that the target organs get innervated by both. So sympathetic innervates the eye, and so does the parasympathetic. If you're looking at things like the liver, the sympathetic nervous system innervates the liver, and so does the parasympathetic nervous system as well. So in other words, all of your visceral effectors have two different branches coming in, one from the sympathetic and one from the parasympathetic, and that's something that I want you to make sure that you know. So nearly all of the organs that we look at that are found inside your body are supplied by both branches, the sympathetic and parasympathetic of the autonomic nervous system. So there's another feature that I want you to be aware of, and let's take a look at the sympathetic first. I told you that they all originate within the CNS. So here's the CNS, here's the spinal cord running down here. Here is thoracic, here is lumbar, the L that you see represented here. Here is the brainstem at the very top. So they all originate in the CNS. So just taking a look at what's happening in the sympathetic nervous system, where do you see the majority of the preganglionic fibers coming from? Are they coming from the brainstem on the left? Yes, no? Are they coming from the brainstem up here? No. So all of these little lines that you see coming out represent the preganglionic axons. And these preganglionic axons, if you're looking very carefully, the first group of axons leaving the spinal cord, they all leave in the sympathetic nervous system at the level of the thoracic sections all the way down to the lumbar sections. So in other words, the origin of the preganglionic neurons that are part of the sympathetic nervous system are all thoracolumbar. They all originate either somewhere in the thorax or at the level of the lumbar spinal cord column. Is that okay for everyone? The second thing that's really important about this is these preganglionic axons that are coming out of the sympathetic spinal cord that come from the thoracic all the way to the lumbar also have really, really short preganglionic axonal fibers. They don't go very long. So they will hit their targets almost immediately as they leave the spinal cord. The next group of neurons, in other words, the postganglionic, are all sitting here. They're sitting in these little round circles, and they don't have to go very far. So these myelinated preganglionic axons in the sympathetic nervous system are not very long, okay? So the preganglionic axons are all myelinated, and in the sympathetic nervous system, they're very, very short. They don't have to be very long. Is that clear for everyone? Okay, so if we take a look at the parasympathetic, a couple of things again to point out to make sure that you are aware of and to make sure that you understand as a general feature. Are they coming out of the same place in the spinal cord? No? Perfect. So they are not coming out of the spinal cord. They're not, the origin is not from the thoracolumbar. They are actually coming out of the brainstem. And so we say that their origin, the preganglionic origin of the parasympathetic, is actually craniosacral. So we either get them at the level of the brainstem up here at the top, or we get them down here at the sacral part of the spinal cord at the bottom. So they are very different in terms of their origin. The other thing to make note of is when they leave their part in the CNS, they will all go out to meet the next group of postganglionic neurons. Again, they're all myelinated, but these myelinated preganglionic axons, do you think they're longer or shorter? So these solid lines that you see here are all preganglionic myelinated axons, and as part of the parasympathetic nervous system, they are indeed much, much longer, okay? So the dashed lines show you what's happening with the postganglionic neurons. So they've reached their ganglia, these groups of neurons here, and now they're going toward their target. They're the postganglionic axons, and these postganglionic axons are all unmyelinated, okay? And these are not myelinated. The preganglionic ones are. It's exactly the same thing here in the parasympathetic nervous system, and you can see that it's the exact opposite. So the preganglionic in the parasympathetic are really long, but the unmyelinated postganglionic axons are really short. Do you have a question? No, not necessarily true. It really depends on what you're looking at, but the response rates, because the overall targets don't tend to be like from the top of your body down to the bottom, they don't tend to be that slow. Okay, so again, if you're taking a look here, the overall postganglionic fibers tend to be unmyelinated before they hit their target tissue, and again, there are some fundamental differences between the sympathetic and the parasympathetic nervous system. So all of these here are releasing acetylcholine, and the ones here are what we're going to be talking about next. So this illustrates again for you in a slightly different way the difference between the myelinated axon. Parasympathetic is longer, sympathetic is shorter. They reach their targets in those little clusters that you saw, those little balls right beside the spinal cord. The sympathetic ganglia are really close to the spinal cord, and the secondary postganglionic axons tend to be much longer, and they're also unmyelinated. The parasympathetic preganglionic fibers are long. The parasympathetic postganglionic fibers are short before they reach their target tissues, and they're also unmyelinated. The other thing I'm going to mention to you here at this point is that acetylcholine is the neurotransmitter up here. Acetylcholine is the neurotransmitter here, but at this level, the postganglionic axon, what it releases in the parasympathetic is not the same as what is released on the sympathetic. And as you can see in the diagram, usually with the sympathetic, it's norepinephrine, and with the parasympathetic, it's going to be acetylcholine. So it's acetylcholine pre and acetylcholine post for parasympathetic, and for sympathetic autonomic nervous system fibers, it's acetylcholine, and then it's usually norepinephrine or epinephrine. Do you have any questions about that before we go on? No. Okay. Again, lots of similarities, lots of differences. So we've primarily been looking at the differences from where they start, so where they originate, so whether it is, again, found in the brainstem, or whether it's found within the spinal cord as their starting point, and the distances traveled by the different types of axons, whether it's sympathetic or parasympathetic, longer or shorter differences. And we'll see that the effectors are very different than what we've looked at before. So I'm going to go through a couple of key points now in the sympathetic nervous system, unless you have any questions, and then we'll talk about what sorts of things the sympathetic nervous system is responsible for. From that first section, though, you should be really 100% clear on the differences in length between the pre and post, the origins for the sympathetic versus parasympathetic, where in the spinal cord or up in the brainstem, and what gets released in the preganglionic versus postganglionic in the sympathetic versus the parasympathetic system. So specifically in the sympathetic nervous system, one of the things that, again, you don't have to worry about memorizing all of this. So when you activate the sympathetic nervous system, when it's fight or flight, in most cases, like the vast majority of cases, when you turn on the fight or flight response, everything gets turned on. Okay, it's not like the parasympathetics. Parasympathetic can be much more localized. You could activate the parasympathetic just in your gut region, for example. But when you turn on the sympathetic nervous system, everything goes on at the same time. It's a generalized response. It's not segmented, or it's not localized to a specific area. So when you turn on the sympathetic nervous system in fight or flight, it might be that you're increasing your alertness, and that certainly happens. You will also increase your energy levels for brief bursts of time. And you can think back to what happened during your midterm, right? Were you guys really excited or jacked up to write the midterm for the first 5 minutes until you saw the first question that you didn't really know the answer to? You probably were. And then what happened to you after you wrote your exam? You probably didn't feel so good, right? You probably felt, I don't really want to go to lecture. I really don't want to stay awake. I want to go to sleep. And that's because the sympathetic nervous system is really great at increasing your alertness, increasing energy. It makes you feel good for brief periods of time. It helps you to increase your respiration rate. You need to breathe in more oxygen. It increases your heart rate and everything else, increases your muscle tone levels, and it gives you all that glucose that you need to get that feeling of energy and euphoria. But for short bursts of time, that's great. If you don't have the restorative actions of the parasympathetic, it actually becomes very, very deleterious for you. So this gets back into the specific things that I need you to know for the sympathetic efferents, everything coming out of the CNS. So the preganglionic efferents that I've mentioned to you previously all come from the thoracolumbar region of the spinal cord. So we would not see the sympathetic nervous system coming out of the sacral end. That's the parasympathetic. We would not see it up in the cervical. Everything that we see in the sympathetic is from either the thoracic part of the spinal cord or the lower lumbar part of the spinal cord. It also comes out of a specific region known as the intermediolateral horn of the thoracic cord in particular. And I'll just show this to you, and I'll come back to this again. And this is that intermediolateral horn. So we talked about the dorsal horn, the ventral horn, and this is where all of those preganglionic fibers from the sympathetic nervous system are sitting. So they will synapse, they will make these short distance axonal projections that are myelinated to a group of neurons, which we call ganglia, that are parallel to the spinal cord. And again, I'll show you that after the next slide. And they sit very, very close, right beside the spinal cord. And some people consider them motor neurons, that they're not true motor neurons. They do not actually cause muscles to contract. And so they're not motor neurons in the sense that even though they release acetylcholine, and they are innervating a target and they come out of the lateral horn, they're not motor neurons at all. So these postganglionic efferents that they innervate and they release acetylcholine onto, it's not always a secondary nerve. It could be a target tissue like a gland. And I showed you an example of a gland in that cartoon a few slides back. And then, and in turn, that gland, when it receives acetylcholine, might release noradrenaline into the bloodstream. And that's part of the adrenal gland response. So it could be an adrenal gland that responds to sympathetic nervous system input. There are two exceptions to this. So when we look at the sympathetic nervous system, when we look at sweat glands, acetylcholine is the neurotransmitter that is being released. It is not noradrenaline that's being released to activate these sweat glands. We won't talk about this in any detail. The acetylcholine at sweat glands, I want you to know. But for muscle vasodilation, which I'll leave for Dr. Key to talk to you guys about, again, it's acetylcholine that causes muscle vasodilation at the blood vessel level in these different responses. So all I want you to get out of this is that it doesn't have to always be a postganglionic nerve. It could be a target tissue like a gland, the adrenal gland being one example, and that it doesn't always have to be noradrenaline. And the only example I want you to remember is that it's acetylcholine that might be released onto the sweat glands, even though it's still the sympathetic efferent. And again, it's this overall structure. It's coming out of this intermediate region in the thoracic or lumbar region of your spinal cord. It sends its axon out, and it will send its axon out a short distance, and this is that short distance that we looked at earlier before it makes a synapse onto the postganglionic neuron. And this secondary postganglionic neuron usually releases noradrenaline, but not always. And it doesn't always have to be a sympathetic ganglion. It could be an adrenal gland, and that adrenal gland might release noradrenaline instead. Is that clear for everyone? So it's still acetylcholine here at this synapse, but it's the release of noradrenaline usually, except for sweat glands, in the secondary postganglionic or in the gland that it's innervating. Any questions about this before we go on? Today's the time to ask questions. Like if you're not sure, follow up and ask. I'm sure there are lots of questions, or you might be thinking about it. 100% clear for everyone? No, not sure. Okay, well, we'll go on, and you know, hopefully you'll have an opportunity to think a little bit more about that. So we won't spend a lot of time on this because, again, I'll leave that with Dr. Key, and you don't have to spend a lot of time going through this slide. I won't be testing you on this slide. What I want you to get from all of this is that the sympathetic nervous system is important in controlling blood flow to different muscle regions. It's a more complex phenomenon that we'll be spending time on today. The major influence that we look at in the sympathetic nervous system is all noradrenaline release. And when noradrenaline is released onto muscle and onto other types of blood vessels, one of its major effects is to cause vasoconstriction. And out of this whole slide, that's really all I want you to take away. And what does vasoconstriction mean to you? So it causes the blood vessels to constrict, the diameter gets smaller, and when the diameter gets smaller, what happens to the blood pressure? Blood pressure increases. And so this particular sympathetic nervous system response helps to maintain the blood pressure in many important ways, and we'll talk about this baroreflex response a little bit later that helps to maintain that blood pressure through this noradrenergic vasoconstriction. And we won't worry about this sympathetic cholinergic vasodilation. We'll leave that for later. The main points to take away is noradrenaline, when it gets released onto blood vessels, for example, it will cause vasoconstriction, and this vasoconstriction is really important for the regulation of blood pressure that we'll be talking about a little bit later on in the course. In today's lecture. So again, if you're taking a look, this is that chain of ganglia. You can actually see these round little balls. These would actually be running parallel to the spinal cord that you can't really see well in this particular diagram, but you can actually visibly see these chains of ganglia parallel on both sides of the spinal cord in these types of surgeries that are being outlined for you here. Okay, so in terms of the summary, most of this is what I've already discussed with you. So the origin of these preganglionic neurons, all at the level of the thoracic. T1 is the beginning of the thoracic portion of the spinal cord, all the way down to L2, which is the lumbar region of the spinal cord, and then these sympathetic preganglionic axons will make synaptic connections. Their first connections usually at the level of the sympathetic chain that run parallel, short distances, all myelinated here, releasing acetylcholine, and then depending on what their targets are, they will send out these longer projections out toward their targets that will cause release of noradrenaline at their target organs to produce the responses that we'll be looking at a little bit later on. Okay, so that's the sympathetic. The parasympathetic is somewhat similar. We've already looked at the overview of it. It's a little different in a couple of regards. All of these different sympathetic, parasympathetic nervous system efferents originate in different cranial regions. So it's at the level of the brainstem, and in the brainstem we have these cranial nuclei, those cranial nerves that we talked about, these motor neurons that make up cranial nerve number 3, cranial nerve number 7, cranial nerve number 9, as well as the vagus nerve, all are part of the parasympathetic nervous system. In addition to that, as part of the intermediolateral part of the sacral spinal cord, so it's cranial, sacral, we will have the origins of these preganglionic parasympathetic fibers. So all of these will then project those longer distances. So the myelinated axons project to longer distances to their secondary postganglionic targets, which are usually embedded directly in the organ itself. So the group of neurons, the cell bodies of the neurons are sitting right near the organ. It could be, for example, the SA node of the heart. It would be considered the target for the parasympathetic nervous system. It could be directly into the nervous system of the gut. And here, acetylcholine is the neurotransmitter that's going to be released from those postganglionic axons as well. So as they reach the SA node, at the SA node, they will be releasing acetylcholine. And have you guys in your tutorials talked about the vagus nerve at all? Yes, no? So if you were to take a look at the cranial nerves, they all start off in the brainstem, and from the brainstem they run different distances. Some will run up to the eye, some will run up to different regions in the face, some will stop around your neck, but others, like the vagus nerve, actually innervate everything in this part of your body. Everything in the thoracic to the sort of abdominal region is innervated by the vagus nerve. So it's a huge nerve. It's a very important nerve that you'll hear a lot about in the second half of the course when you start learning about the digestive system. It's really under the control of the 10th cranial nerve known as the vagus nerve. And if I'm not mistaken, this year you've also learned the nitric oxide pathway with Dr. Key as well? No? Okay, so we'll go through this really quickly. Usually he covers it. I'm not sure. Every year it tends to be a little bit different. So I stuck this slide in just as a potential slide that we can go over. Since he hasn't covered it, I will tell you ahead of time that I will likely ask you a question directly from the slide that I'm going to show you next. So in the parasympathetic nervous system, not only do we have acetylcholine, we also have nitric oxide as a as a neurotransmitter. It's a gas, but it can also be used as a neurotransmitter. And one of the things that this parasympathetic nervous system neurotransmitter, when it gets activated, one of the things that it does is it causes vasodilation. And it doesn't cause widespread vasodilation through your entire body. One of the places where it causes vasodilation after stimulation is to cause vasodilation, especially within the penile region. So this is a potent vasodilator in one region of the body. As I told you before, unlike the sympathetic, this has local regional effects. It's not released from vesicles, so it's not encapsulated in vesicles at all. It is a volatile gas. As you start to form it, from L-arginine being converted in the presence of calcium. So it works just like any other neuron. Calcium comes in after an action potential gets generated. As an action potential goes down into this parasympathetic nerve, calcium influx to those voltage-dependent calcium channels will activate an enzyme found in these parasympathetic neurons that innervate the blood vessels within the penis to cause nitric oxide synthetase to become activated. And when it gets activated, it will cause L-arginine to be converted into nitric oxide. And why nitric oxide is really important is that it can then cause, it can cross over to its, into the blood vessel, and because it can cross over really quickly, it can cause the inhibition of guanylate cyclase, or the activation of guanylate cyclase, to convert GTP into cyclic GMP. And this cyclic GMP is really important because it activates various metabotropic receptors that are important for vasodilation to occur. So this whole cascade is really important because nitric oxide will cause the increase in the cyclic GMP, which then acts on metabotropic receptors that are allowed to cause vasodilation to occur. So all of this occurs in a neurotransmitter-like fashion, but it is not a true neurotransmitter because we don't store this in vesicles. It's just stored as an inactive amino acid in the presynaptic terminal. So is that clear for everyone? Okay, so this part makes it look a lot like a neurotransmitter. You still get calcium calcium entry through voltage-dependent calcium channels presynaptically in this parasympathetic neuron. It activates nitric oxide synthetase, and this causes the conversion into nitric oxide, and then we need to start forming cyclic GMP. And we won't worry about the phosphodiesterase inhibitor that normally causes the reverse reaction. Okay, so in terms of what the overall effect is, when we take a look at the sympathetic versus the parasympathetic, when we activate the sympathetic, we might have increased cardiac output, increased heart rate, increased stroke volume, increased effectiveness of the heart, in other words. And then we might have skin vasoconstriction, we might have sweating, we might have piloerection, your hairs on the skin will stand up straight. You might have blood diverted from the gut and then diverted from the skin, all directed toward the muscle. In the parasympathetic, it does almost the exact opposite. However, we don't look at them as being antagonistic. We look at them as being complementary. When one gets turned on, the other one kind of takes a break, and then it makes sure that the other system, what it's been doing, kind of gets replenished or refreshed. Okay, and what we're going to do next is take a look at how all of these different groups of neurons can be put together in more functional reflexes that are autonomic reflexes. We've seen one example of an autonomic reflex important previously, and why these autonomic reflexes are important. But I'm hoping that now that you know the structure and you know that it's coming from the spinal cord, it looks like a reflex coming out of the spinal cord, and that we can start building these reflexes into some more complex networks, just like we've done previously. Again, before I go on to that, and I'll just mention this in passing, I will not be asking you a specific question from this slide. Your intestine, the intestinal wall, especially the small intestine, actually is a really important secondary nervous system. You have all of the neurotransmitters that you have in the brain So there's somewhere between 50 and 60 neurotransmitters. You have 50 and 60 neurotransmitters represented in the gut as well. And that's all under the control of the autonomic nervous system. And you'll learn more about that in the second half of the course in PSL 301, where you'll learn about how the parasympathetic nervous system will control this group of nerves that are embedded directly within the walls of your gut, and why that's important for gut motility. And you'll learn all about that later on. But I won't go into that in detail here. It's just to give you an overview. And when you get to that section, you'll think back to this parasympathetic causing this rest and digest, and you'll remember how this works in terms of the overall manner in which the acetylcholine versus noradrenaline works on this enteric nervous system. So I'm going to go through one reflex that we've already seen before as an autonomic reflex. You don't have to think about it. And in addition to that, you also will make use of all of the circuits that I've just outlined for you. So it's the same thing. It's a reflex, and it's part of this autonomic circuit that I've already described for you. And hopefully now you'll understand how some of these different reflexes come into play. So we've talked about this pupillary light reflex previously, right? Yes? Yes. Okay. Do you guys remember how it works? Yes? You will need to remember how it works for the upcoming term test. You'll also have to know it for the final exam as well. So it's a good time to reestablish the importance of this reflex. So this is what we're talking about. In response to light or in response to darkness, your pupil either gets really large like you see here, or it gets really small if you're in light. And we've talked about that before. What's going on over here on the left? So again, really simple question. Is it a constricted pupil or is it a dilated pupil? Okay, great. So a dilated pupil. What causes you to have a dilated pupil like that? And again, think about this. You're shining light to be able to take this picture. And is her pupil dilating? It's still dilated. So what causes that? Sorry? Atropine? Sure, it could have been atropine. Amphetamine? Maybe amphetamine. Okay, let's see if I actually have this on here. But when you get pulled over by a cop, and they are actually taking a look at your eyes, they want to see a couple of different things. One is, do you have like this type of response on the left? I happen to know because my brother's a cop up in York Region. I'm allowed to speed in York Region as a result of that. And one of the things that I've was able to ask him a little bit about, like, you know, when you pull people over and you want to see if they're drunk, like all those different types of reflex activities, what other things do you kind of look for? And one of the responses is to see if, for example, you have this really massive dilation. And if you do, you might be on different types of drugs, right? So behaviorally, you might be okay, but if your pupil is really dilated, because cocaine is actually a noradrenaline reuptake inhibitor, if you're on things like cocaine, you can have this type of pupillary response, and you will be fully dilated even in the presence of light. And so when the police officers actually pull you over, they are looking for specific things, and these are one of the things that we can talk about in terms of this autonomic reflexes. So a lot of stimulants like cocaine, potentially amphetamines, amphetamines work in a slightly different way, cause this chronic pupillary dilation. So I want you now, when we start going through this, to start revisiting different aspects of the course. So this is a good way to know how much of the course you sort of remember. Those retinal ganglion cells that we talked about, those million or so retinal ganglion cells that leave the back of your eye, those retinal ganglion cells also, some of them have a light absorbing pigment that we've talked about previously, known as melanopsin. And what melanopsin is really good for is determining luminance levels, like how bright or how dark something is. It doesn't really do much more than that. It's not a photoreceptor. Remember, it's a ganglion cell, and this ganglion cell itself has a light absorbing pigment known as melanopsin. This is the beginning of this pupillary light reflex, and it's also the beginning of the circadian rhythm that we'll be looking at as a higher level autonomic function a little bit later on. These same melanopsin containing retinal ganglion cells are important for synchronizing your response to the day-night cycle, and whether or not you should be falling asleep at 1 o'clock in the morning. All of you should be, but in fact, the majority of you are not, right? And why are you not falling asleep at 1 o'clock in the morning? Yes, you're watching Game of Thrones, absolutely. So that's one reason. But another reason is you guys stare at screens an awful lot, and that screen that has a blue-green tint to it is affecting your circadian rhythm by directly affecting these melanopsin-containing retinal ganglion cells at the back of your eye. And we'll see that a little bit later on today. So just a reminder about the two different muscle systems that cause your pupil to constrict here on the right versus your pupil to dilate here on the left. And again, we have these two different types of circular versus radial muscles. There's a lot on this particular diagram, and I'll be completely honest with you. I'm going to ask you a question related to this diagram, and I'm going to be asking you questions related to what's written here on the left on this diagram as well. And I need you to know the circuitry. I need you to know what is being activated in the sympathetic nervous system, and I need you to also know what is being activated in the parasympathetic nervous system. I also need you to know and recall, because we actually talked about this in the last lecture, is one of the important factors is that this pupillary light reflex, and we talked about it again in passing in the lecture on vision, that this pupillary light reflex, when you shine light into one eye, both pupils constrict because it's organized in this midbrain tectal area. And this tectum is part of that superior colliculus that we looked at for tracking things in space and for being able to coordinate how your eyes and head move in the same way. So this pupillary light reflex, as any type of reflex, requires some sort of sensor, and the sensor is that melanopsin containing ganglion cell, and it uses those on and off afferents that are arranged through those melanopsin containing retinal ganglion cells to act as a luminance detector. Is there a lot of light or is there a lot of darkness? And it will send axons back into this pretectal nucleus, and this is called an olivary pretectal nucleus, and it's still part of that midbrain, it's still part of that superior colliculus. And what happens is that when this starts to go and get sent back into this pretectal area, if it's too bright, if these luminance detectors, these on-off afferents are saying that there is too much light, it will activate the parasympathetic reflex, and this parasympathetic reflex through the third cranial nerve back up to the ciliary ganglion will then cause the circular iris muscles to constrict. It will cause those to constrict, and overall it will cause the constriction of the pupil. The pupil itself gets smaller. And because it's parasympathetic, we are going to be using acetylcholine in this response, okay? And you need to be clear on that sort of neurotransmitter as well. So the third cranial nerve will activate and cause the constriction through the release of acetylcholine of that circular sphincter, and when that circular muscle contracts, it will cause the pupil itself to constrict when there is too much light. Too much light, parasympathetic, third cranial nerve, activation of the circular muscle through the release of acetylcholine. If it's too dark, the melanopsin containing retinal ganglion cells that detect light levels in your eye, if it's too dark, it activates through neurons that are found in the olivary pretectal nucleus. It will activate a sympathetic response, and this sympathetic reflex will travel all the way back down into the thoracic cord. So remember where the sympathetic nervous system is. Okay, the sympathetic nervous system is not part of your cranial nerves. It's not in your brainstem. It actually has to go all the way back down to the thoracic level of the spinal cord, and then from the thoracic part of the spinal cord, it has to go all the way back through the sympathetic chain right next to it to the radial muscles back to your eyes. So is it a longer route? Yes, it is. It's a much longer route than is taken by this parasympathetic pathway. It will cause that those radial muscles to constrict, and it will cause this dilation of the eye of the pupil overall because of the actions of noradrenaline or norepinephrine. So I want you to be clear on this reflex that the sympathetic system will activate through the release of noradrenaline, the radial muscles in the pupil, and when the radial muscles contract, overall it causes the pupil to dilate. So if noradrenaline is being used here and it causes dilation, and noradrenaline causes dilation, and you know that cocaine stops the breakdown of noradrenaline, now you know one of the reasons why in an individual taking lots of cocaine chronically, why their pupils are dilated all the time. It's because they can't break down this noradrenaline. This response is always being activated. It's like having this sympathetic reflex all the time, even in the presence of light. Okay, so I think here's a good place to take a break. I think based on the number of slides I have left and how much time it's taking to go through them, I'm probably going to finish just after about 7:15. I have a couple of practice questions for you at the very end, but if you do have a recorder up here, please don't leave and then come back at 8 o'clock, because I'm going to take those, I'm going to sell them somewhere, I guess. But if you can stick around, I'm not planning to go right up until 8. It will probably finish around 7:15-ish or so. And we'll take a break right now. Hello.
Hi, I have a question from last week's lecture.
Okay.
When talking about senses like taste and smell, we talked about like primary neurons, especially, but I was wondering if the primary neurons are the receptors, or is there like specialized cells that gets the stimulus and that sends to the primary neurons, or is the primary neuron itself a receptor?
No, so there are, in some cases, there are the taste buds, for example, or the olfactory epithelial cells that act as the transducing elements. So they take a chemical signal, they release neurotransmitter onto the neuron that then sends a signal into the CNS.
Okay. Is the action potential always trigger at the primary neurons, or is it the primary neurons? So it has to be. So always the primary neurons. Okay. I have one more question. You said about noradrenaline always the piece of consumption. But I think because of you we learned how it can cause these fibers as well. So like for this course, do we just know that it's but do we have to know which part of the body?
I was wondering, could you post your email that you got in the first slide?
It's on every lecture.
Really?
It's on every lecture.
Um,
On the bottom left corner.
oh,
It's
okay.
got
Okay.
Gmail.
Second question. So, do you actually have office hours on
I'm not sure yet.
I'm not sure.
We do have like a tutorial, so I'd rather try to answer all the questions that everyone has at the same time. The question would repeat the answer.
Okay.
Just making the assumption that most people study till the weekend might be erroneous, but if you have questions that are really short and concise, then email me on this email.
I just emailed to the.
Professor, my question is, why is it that ANS is more segmented than the SNS? So why is it that when you turn on the sympathetic nervous system, everything turns on, and then the parasympathetic, why is it more segmented so that you turn on one part of it, but not the rest?
it's more about the brain as well as the specific circuits and parts inside.
I'm sorry, when he looks like this whole part is like freaking out. I started when that doesn't matter. This part, so it's saying that there doesn't always have to be a face scan. It can be a yeah. Oh, yeah, the nitrogen. No, nitric oxide. It's not a true neurotransmitter. It's more understanding of the
I have two quick questions. First one, when we say cholinergic, it does means that it's the use of acetylcholine in both post-synaptic and pre-synaptic. I mean the post-ganglionic levels. Pre-ganglionic in both parasympathetic and sympathetic, or just okay. And then second question, when you say that when you mentioned focus, what's the memory is between the basic and structure? So it confuses me. When it's a basic and structure, that's why you see the hole in the nose, right? When we check for okay, this is okay. You also mentioned alcohol. Is it the same way? Is it dilate or is it the opposite? Okay, it's not the business. Okay, makes sense.
Hi. So in the sympathetic, you know how you said that it had to go all the way down to the thoracic and then all the way back up, so it was kind of like a longer route. Does that mean that your pupils would dilate, I guess, like slower in comparison to like how they would constrict in the parasympathetic? How come, like, I guess in younger people, it's around, I guess, the same time you could say, even though it has to go through like a longer route, I guess, to dilate in the sympathetic.
Um, I think it has very little to do with the actual distance. It's really not that
Yeah.
much. So thoracic is down here.
Yeah.
The other one goes down to the not that much, but the efficacy of neurotransmitter release does change as you get older.
Okay. Alright, thank you.
Okay, so I'm going to go through some higher orders of different autonomic control and how the autonomic nervous system gets integrated in a number of different higher order levels of reflex loops, if you want to think of them that way. We'll be talking about different structures, but it's the same overall idea. We have things that can happen at the level of the spinal cord, and we went through some of the circuitry at the level of the spinal cord. We will be talking about the different parts of the brainstem and the brainstem organizing areas, and then we'll talk about this master integration area known as the hypothalamus as all part of this autonomic control. There is some input as well as control at this level of the hypothalamus in terms of the limbic system, and we talked very briefly about the limbic system previously, and we'll revisit the limbic system because, as you know, if you're really scared or you're really excited or you're really angry, it can have a response in terms of the autonomic nervous system, whether or not you want it to happen that way or not. So it could be also really voluntary. You could imagine, for example, like the most scary experience, and if you revisited the most scary experience that you have using the higher brain centers, the highest brain centers can actually come back down the other way from the cerebral cortex, activating the limbic system, and then you might actually start showing a fear response, right? So you're no longer in danger, but if you're recalling that and under the right situations, this can be sort of a voluntary activation of the autonomic nervous system. Some people, I haven't encountered them myself, can actually start trembling or start crying because of their recall of a really scary or a really traumatic situation, because they're voluntarily activating this higher level limbic activation of the hypothalamus as part of the autonomic system. And again, there's some instances that people can learn to activate this higher level control to control blood pressure. And some individuals have used this biofeedback process where you're looking at a blood pressure monitor, and you're watching it, and you want to lower your blood pressure, and this form of biofeedback where you're consciously causing the autonomic response that controls your blood pressure to lower your blood pressure, sort of as an effective way to modify or regulate this autonomic activation in this sort of hierarchical manner. So conscious control, then emotional control, and then this area of the brain, the hypothalamus, is where we're going to be spending a lot of time. So related to these lower levels, at the level of the brainstem and at the level of this reticular formation, we have a group of neurons that are really important. And these groups of neurons are clustered together in the brainstem as part of that reticular formation into these brainstem autonomic centers. So there are going to be some sympathetic modulatory tracks, so groups of axons coming from the raphe nuclei, which release serotonin. So any of the raphe nuclei that we encounter will release serotonin, and the locus coeruleus might get some sympathetic innervation. And when the locus coeruleus gets activated, we'll see that the noradrenergic system comes into play. Many of these brainstem autonomic centers that are found at the level of the brainstem involve controlling the cardiovascular rate, how rapidly you breathe, the respiratory pattern. And again, it's sort of like looking at those CPGs that we looked at in the spinal cord. We can look at the same sort of CPGs here in these autonomic centers as well. Another thing to keep in mind as sort of a symmetry with what we looked at before, there's another region known as the periaqueductal gray. Sometimes you'll see it in short form as PAG, and this periaqueductal gray region is sort of similar to the midbrain premotor center that we looked at before. So complex types of behaviors that move all of these different autonomic functions in the same way. So things like feeding responses, you don't have to think about chewing your food. As soon as you see it and you're hungry, these types of autonomic behaviors, when you're hungry and you eat, this feeding behavior is pre-planned in this periaqueductal gray region. For example, vocalization during feeding or during things like sex, all of that is organized at the level of the preaqueductal gray. And it's sort of like having a premotor center similar to what we've seen before. So many of you will be modifying all of these brainstem autonomic centers, and you probably do that without being aware of it. Can you give me examples of how you are changing the activation of some of these different brainstem autonomic centers? What do you do? Nothing? Yes? So drinking energy drinks would be one way. You drink coffee. All of those things, by the way, modify your responses. All of the modulatory systems that we're going to be looking at in the next few slides get modified every time you drink these different types of energy drinks. What is in these energy drinks? Caffeine. So why is caffeine good for you? Keeps you alert. Okay, why does it keep you alert? What does caffeine do to your brain? It stimulates it. How? It's like, it doesn't work through magic, right? It stimulates your brain, absolutely. Keeps you alert. Does all those things that we've talked about previously. So of the two different systems that we looked at, is it going to be more likely that you're going to be activating the sympathetic or the parasympathetic? What do you think? Sympathetic. What's the neurotransmitter that gets used by the sympathetic nervous system? So it's noradrenaline. One of the things that modulates the activity of noradrenergic neurons is a substance known as adenosine. And as you get more and more tired, and as you get after exercise, you build up a lot of adenosine in places like your brain. And adenosine actually turns off the neurotransmitter noradrenaline. And so what caffeine does, it actually antagonizes the adenosine receptors. So it's like not having any adenosine around. And so caffeine helps you to pick up the release of norepinephrine that way. And we'll talk about that a little bit later. So these next few slides are going to be really important in terms of a couple of things that I'll highlight for you. And please make sure that you're aware of the different systems as I highlight them for you. So we're going to be looking at in the brainstem and in different regions of the brainstem, this diffuse collection of nuclei. So it's like the reticular formation that we looked at before, but we're going to call it the reticular activating system, a group of neurons and nuclei within the brainstem. These all work on modulatory activity. So they will cause an overall change once they get activated in CNS activity. So for example, we might have 4 different systems. I'm showing you 3 right now. The cholinergic system is going to be really important because it determines how aware you are of something, how alert your brain is. And if your cholinergic system is activated, your attention levels are going to be going up. And it will be an ascending system. It will go from the pontine reticular formation into higher levels in the brain. I'll outline what that means for you in the next few slides. The serotonergic system is going to be important when you are looking at stressful situations. It's a very powerful mood elevator and depressor if you don't have the right levels of serotonin, and it will also influence the sleep-wake cycle. The adrenergic system, as I mentioned, is important for stressful situations as well as for vigilance. The last two, serotonin as well as adrenergic, both of these are going to produce actually strong motor effects. It's not going to make your brain get stimulated. It's also going to cause your muscles to become activated as well. So if we start looking at the anatomy of the different systems, and again, it's more important for you to pay attention to what's up here than down here on the bottom. So when we look at the cholinergic system or the acetylcholine system, there's a couple of things I want to really point out for you. So one is this begins at the level of the brainstem, and here is the brainstem down here, and here is the pons down here, and here is the rest of the medulla down here. So in this area of the brainstem, we have a group of neurons that come together that produce what is known as the pontine reticular formation. And you'll see that some of the neurons in the pontine reticular formation, this loose network of neurons that release acetylcholine, some of them are descending. You'll see some of the projections going down, but the majority of them are actually ascending. They will send their fibers up higher in the brain, which is what that slide that I just showed you was indicating. So it will send their projections up to other cholinergic neurons like the medial septal nuclei, or it might send projections up to the basal nucleus of Maynard. And again, from there, they will also send cholinergic outputs to the other areas of the brain. So when you want to stay alert, when you want to be as alert as possible, you turn on this acetylcholine pathway from the reticular pontine formation, and will activate the entire brain through these ascending projections. Do I want you to know what the nucleus uh, of Maynard is, basalis of Maynard? Yes, I do. I want you to know that one. And I also want you to know that this medial septal nucleus is also an acetylcholine releasing group of neurons that is part of this acetylcholine system that is used in being awake and being alert. Do you know why those two groups of neurons that release acetylcholine, that receive input from the reticular pontine formation, why are those important? They're really important right now in physiology, pathophysiology, and neurodegeneration. Do you have any ideas why individuals are looking at this? So yes. For Alzheimer's, exactly. So one of the things, again, you look at Alzheimer's, the whole brain is starting to atrophy. And in Alzheimer's, there's a big issue with being able to pay attention and to be able to direct your attention. You tend to be forgetful. And some of that is because these two nuclei that are cholinergic, that cause you to be alert, to pay attention and everything else, in Alzheimer's disease, these are some of the first neurons to actually go. And if you're not able to pay attention and you can't be alert, you can't really answer questions well. You can't really know where you're at, and sort of that forgetfulness sort of follows as a result. And again, we won't talk about tricyclic antidepressants this year. We'll just look at the serotonergic system, and you can see that it's a very different looking group of brain autonomic centers. It's not like this pontine reticular formation. Here you'll see that the groups of neurons that produce serotonin is much more widespread, and this is the Raphe nuclei. All of these red blobs that you see here are the Raphe nuclei. These Raphe nuclei are important because they will also send projections into the cerebellum. So the cholinergic system, for example, does not send projections into the cerebellum, but you'll see that this serotonergic system does, and you'll see that it's also ascending. It sends it up into the entire cortex, into different regions of the cortex as well. And again, the major thing here is the Raphe nuclei tend to be much more broad, distributed, and will also send projections into places like into the cerebellum. And in addition to that, I'll just highlight this for you now, that they also send projections into the hypothalamus. The third system that was on that original slide was this norepinephrine system. And again, it involves this one group of neurons, sitting right here in the brainstem. The locus ceruleus, or the blue location, will send axons up to the entire cortex. It will send projections into the cerebellum. It also sends projections down to the brainstem. But this is where all the noradrenaline in your brain, we're not talking about being released in the spinal cord, but in your brain, when you need noradrenaline, it's this group of neurons that releases noradrenaline to all the different parts through all the axons that are being shown here to various parts of the brain. So this locus coeruleus is what starts this whole process off. The one group that we didn't have on that original slide that's really important as well, again, keeping this all clear, is this group of histamine containing, histamine synthesizing, histamine releasing group of neurons. And this histaminergic system starts off in the posterior part of the hypothalamus. And we'll see why the hypothalamus is going to be important in a few moments. And in a region of the hypothalamus that is known as the TMN, tuberomammillary nucleus, the TMN, this TMN is going to contain a lot of this histamine. This histamine pathway is going to be really important when we look at sleep. It controls a lot of things related to your sleep-wake cycle, for example, that you may or may not have been aware of. So it starts off here in the TMN of the hypothalamus, and it will send its projections into the forebrain. It's ascending, in other words, just like all the other systems that we looked at. Is this system also descending? Yes, it is, because it also sends projections down into the spinal cord, so it descends from where it is. And again, we'll see that this is really important in sleep-wake control, and this is important when you need to be able to stay awake. And I'll just highlight that for you now. Hopefully by the end of today's lecture, you'll understand why histamine is really important for you to be able to stay awake. How many of you take or have allergies? Some of you have allergies. How many of you take medication for your allergies, like over-the-counter medications? What's usually in your over-the-counter medications? So antihistamines. What are one of the side effects that's on the label for these? Drowsiness. And now, hopefully, you'll start to see why having an antihistamine that blocks the activation of histamine, which is important in keeping you awake, why blocking this might actually make you more drowsy and put you to sleep. Okay, so I'm going to switch gears just a little bit. I'm going to tell you that we have a number of central pattern generators, and we talked about the reticular formation a little bit earlier on. We have some central pattern generators that help to regulate and organize a number of autonomic feeding behaviors, and we're going to look at some of these feeding behaviors, fear reactions, etc., that are part of this autonomic pattern generation. It's that periaqueductal gray that I mentioned to you at the outset when we came back from break in the midbrain that is the most important organizer for these autonomic pattern generators that exist elsewhere in the brain stem. So most of these, not all, are involved in homeostasis, and that's what we're going to be concentrating on for the next 20 minutes or so. And I'm going to take you through the hypothalamus, the most important integrative part of this autonomic pattern generation. So this is what starts and integrates all of the pattern generation that we're going to be talking about, and why the hypothalamus is so important for homeostasis. And along the way, we'll be talking about the limbic cortex and how it regulates the hypothalamus as this autonomic pattern generation occurs. Again, don't bother trying to memorize this slide. It's not that important. You'll, again, pick up most of the points related to this as we go along. When you're talking about homeostasis, it's really a limited number of things, right? So it's your core body temperature. The hypothalamus is important in regulating your core body temperature. It's your blood glucose levels, and the hypothalamus is really important in doing that. And it's important for blood pressure control, and your hypothalamus is really important for that as well. There are a number of sensors that are located within the hypothalamus to detect changes in temperature, changes in blood glucose, and changes as well as osmolality. So how much salt or how much potassium you have in your blood as it goes by the hypothalamus. So when there are changes beyond the norm that it expects, there are a number of corrective actions that get taken autonomically without you thinking about them. That could be anything from release of hormones to a somatomotor response. You might actually have your hair standing on end, for example, as a somatomotor response if you're feeling cold, or you might be trembling as a somatomotor response if your core body temperature has dropped. So the hypothalamus regulates all of these different things by interacting with some of those brainstem centers that we just looked at and some of those autonomic centers that we just looked at, and to turn them on and to regulate their overall function. If you were to take a look at why it's capable of doing so, and again, don't bother trying to memorize this slide. I'm not going to give you a slide that you'll have to label or anything else. If we're looking at the hypothalamus, and here are those organizing centers, that periaqueductal gray. Here are some of those diffuse modulatory systems like the Raphe nucleus, one of the Raphe nuclei, the locus coeruleus. Here is that reticular pontine formation that we looked at. Again, the noradrenergic system, the serotonergic system, etc. All of these things are really close by to where the hypothalamus is located. And we'll be talking about each of these different nuclei in turn down the road. And why is it endocrine related? Because the hypothalamus is sitting right on top of that pituitary gland that you'll be looking at in the second half of PSL300. So the hypothalamus has a number of different roles. It has a number of specific inputs into controlling homeostasis, and we'll talk about each of these in turn. So we'll talk about the nucleus tractus solitarius. So this provides information about blood pressure as well as how distended your gut is. There is another region that sends information up to the hypothalamus, like the reticular formation. This receives information from cranial nerve number 10, and this is really important for relaying information about your skin temperature and comparing your skin temperature to your core body temperature. The retina will tell you a lot about circadian rhythms and the day light cycle, and it does so through the suprachiasmatic nucleus, and you'll see that abbreviated later as SCN. And this is going to be important for circadian rhythms that we'll talk about at the very end, and we'll talk at the end of this section about the limbic and olfactory systems and how eating behaviors and other things can be activated and modulated by inputs into the hypothalamus. So this is a really busy looking slide that I know that you don't have. You don't really need to concern yourself about it too much. Just like the thalamus, the hypothalamus, it has a number of discrete nuclei. So it's not just a small structure. There are a number of discrete regions that are found in the hypothalamus that control things, for example, like the paraventricular nucleus, feeding behaviors. And there are other areas that are involved in causing cooling. There are other areas that are involved in causing, for example, changes in blood pressure. There are other regions that are important in detecting circadian rhythms. So this small region of your brain that you see highlighted is not contiguous. There are certain groups of neurons that are highlighted in different colors that are responsible for different types of autonomic types of behavior. So one of those types of behaviors that we'll take a look at, and this is a slide that I want you to know, this is a slide that you'll likely see either on the term test or on the final exam. As one of those homeostatic autonomic behaviors, one of the most important things is really temperature regulation. So if skin temperature information is different from your core body information, the thermoreceptors at the base of the hypothalamus, at the posterior region of the base of the hypothalamus, will detect any changes between what is going on the outside of your body, as well as what's going on in the blood flowing past the hypothalamus. So it's always monitoring what's going on here on your skin versus what's going on inside your body. So these thermoreceptors at the base of the hypothalamus become really important. So for whatever reasons, and it's not really clear why, it looks like we are more sensitive to actually being hot than to being cold. We have more neurons within, or more thermoreceptors that are sensitive to changes in heat. So if our temperature goes up too much, we have more neurons that are responsive to being hot than we are to being cold. It's more likely that you will have a homeostatic response by raising your core body temperature by 1 degree, then by lowering your core body temperature by 1 degree. And we don't really understand why that is. We think it might be because, you know, a fever can affect a number of other processes in your body, but we don't know why the number of thermoreceptors is very, very different that controls thermogenesis. So if you're too cold, one of the things that happens is that you will start initiating vasoconstriction in your skin. All the blood vessels in your skin close down. You will initiate piloerection. So piloerection, all the hairs on your skin will start to stand up and try to trap as much air in that region as possible. You will actually decrease your cardiac output. So you don't want the blood to flow through your skin as much as it normally would, because that is a way for heat to be released into the environment. There is a shift of fluid out of the plasma. You will start a shivering somatomotor response, and this is all taking place because you have activated the thermoreceptors that are found in the posterior part of the hypothalamus. On the other side of things, if you're too hot, you will initiate skin vasodilation, you'll start sweating, there'll be increased cardiac output. You want to cause as much heat being released from your blood as possible. It will shift fluid into the plasma from the extracellular space. Again, you'll learn about antidiuretic hormone a little bit later on in the second part of this section of the course, and you're going to start looking at the prevention of the loss of fluid through urination through ADH release. So again, we won't worry this year about the different types of input in here. I just want you to be familiar with the different regions of the hypothalamus that respond to cold or respond to heat, and why and how this basic mechanism works, having central thermoreceptors in the base of the hypothalamus versus ones that are on the skin. So the other thing that the hypothalamus is really important in doing is maintaining blood pressure. And blood pressure is really important for a number of reasons. It perfuses your organs, it makes you maintain consciousness if you're standing up, and all of that is occurring here at the level of the cardiovascular center. So this is actually all the way down in the brainstem. It will send some projections up toward the hypothalamus, but this is outside of the hypothalamus. This is in the brainstem. It's in the ventrolateral medulla, and this cardiovascular center, as part of the brainstem autonomic center, is important for regulating your blood pressure. And that's the key thing that I'd like you to take away. Your blood pressure regulation occurs in the brainstem at the level of the VLM, or ventrolateral medulla, and this region of the ventrolateral medulla has a number of neurons known as the nucleus tractus solitarius, or the NTS, and I will abbreviate it and give you the long form on a test if you ever see it. So the NTS receives input from baroreceptors, pressure receptors in other words, near your heart, and it will send information up into the VLM region through this nucleus tractus solitarius. So this information is coming in, and then this information is being relayed into this ventral lateral medulla, and this is where things happen for your blood pressure control. Just like we learned about the different central pattern generators, this is a common theme here in the autonomic nervous system as well. When you activate one group of neurons, the other group of neurons turns off. Okay, we looked at that when we looked at flexor extensor CPGs in your spinal cord. When the flexor group is on, the extensor turns off. When the extensor turns on, the other one turns off. And it's exactly the same thing here. In this situation, it's not two groups of neurons, it's really one long group of neurons, which is known as the VLM. Do you guys know what rostral refers to? So imagine this ventrolateral medulla as a long loaf of bread. And this long loaf of bread has a head region and a tail region. The head region, the one that's closer to the head, if you were to orient it upwards, that loaf of bread, that head region is known as the rostral region, the part that's closer to the head. And the opposite side is the caudal region. So when you see things like caudate, it means tail, and caudal also refers to the tail portion of it. So the front half, or the rostral half, will excite sympathetic efferents. So when information is coming in through the nucleus tractus solitarius and it innervates the front half of the neurons that make up the ventrolateral medulla, it will then cause the ventrolateral medulla neurons in the front half to excite sympathetic efferent axons, and these sympathetic efferent axons will then cause an increase in blood pressure, and we call this physiological response a pressor response or a pressor function. So if your blood pressure has been really low, this is how it would be turned on. So can you think of a physiological way in which you use this pressor function every single day? When is your blood pressure really low, and when do you need to activate it to increase your blood pressure? So when you wake up, yes, absolutely. But what specifically when you wake up? So when you kind of open your eyes and you're kind of hitting the alarm clock, is your blood pressure necessarily increasing? You don't really need it to increase. Probably when you're rolling out of bed and then you're trying to stand up. As soon as you go from being horizontal to being vertical, there's this change in blood pressure. And that change in blood pressure, unless blood goes up to your head now, you're going to faint. Some people have that. And without this pressor response, where the nucleus tractus solitarius has received information from the peripheral baroreceptor saying that, wow, you know what, I suddenly had a big decrease in blood pressure, I should start activating the rostral half of the ventrolateral medulla, we should activate the sympathetic efferents, and the sympathetic efferents should increase blood pressure. Without that response, you will faint. And it goes exactly the opposite way. If it receives innervation on the caudal half actually inhibits the rostral half, the one that increases blood pressure. And as a result, when the caudal half gets activated, it will actually cause a depressor function where the blood pressure starts to drop. So this baroreflex is a really important one for you. So the baroreceptors that we've been talking about are found in the heart, in the carotid sinus, as well as in the aortic arch. So it's part of the aorta as blood leaves and as blood gets pumped out into the aorta. These baroreceptors send their axons up collectively into this nucleus tractus solitarius, or here it's denoted for you as the nucleus of the solitary tract. It's exactly the same thing as NTS. It will signal to the ventrolateral medulla and will increase that sympathetic output. And I'd like you to be familiar with the ventral half versus the caudal half, what is the pressor response versus the depressor response, and that this isn't in the hypothalamus, this is actually in the brainstem region, and this is how we control blood pressure. Do you have any questions about that, or is that clear for everyone? So this is the second homeostatic response that we looked at. This is actually a really complex one as well. And I have a few slides to go through you with on the regulation of feeding for homeostasis. It's important, it's very important for you to maintain your blood glucose. For those of you, and they're probably in a class this big, there are probably one or two of you that have diabetes, and you know that the regulation of your blood glucose levels is really important. If it drops too low, you'll faint, and you'll go into diabetic paralysis and other types of things. And so we know that maintaining your blood glucose is really important, and one of the best ways to maintain your blood glucose is to regulate how you are feeding. And how you feed is really determined by a number of different nuclei within the hypothalamus. And I'd like you to make sure that you are clear on this, and I'll show you this in the next diagram, and then on the subsequent diagram, I'll show you other factors that will influence how these different nuclei get activated or turned off. One group of nuclei within the hypothalamus is known as the arcuate nucleus. Okay, that's one subset of the hypothalamus. In the arcuate nucleus, we have one set of neurons that contain a neurotransmitter known as NPY, or neuropeptide Y, and it will activate a feeding drive. You will feel hungry, you will want to eat. When you want to eat, you are activating these NPY containing neurons that will then stimulate these paraventricular neurons that are a separate group of neurons. In this arcuate nucleus, we also have another group of neurons as part of that nucleus that don't use NPY, that use melanocortin, that actually will activate a different behavior. It will actually stop you from eating. So these are antagonistic groups of neurons, one NPY containing, another one melanocortin containing, and they're both found within the arcuate nucleus, which is found as part of the hypothalamus. So there's another level of control within the hypothalamus. Neurons in another group of nuclei, known as the ventral medial nucleus, act as glucoreceptors. So I told you that when you have low blood glucose, that you will potentially faint or you will run into all sorts of different problems. How does your brain know that you have low blood glucose? Just like the thermoreceptors at the base of the hypothalamus monitoring temperature, you also have receptors that will monitor the blood glucose every time blood flows by this area of the brain. And these will discharge in relation to the ambient glucose levels. Whether you have too much or whether you have too little, they'll change their firing rate. Their overall response is to inhibit those NPY-mediated drives to feed. So I'll also mention in this particular slide that we have a number of hormonal controls on our regulation of feeding response. So one of these is leptin. Leptin is secreted by white adipose tissue. So if you have white adipose tissue or fat, this actually is released by your fat cells, and whenever you're releasing fat cells, whenever you're releasing leptin from fat cells, it produces a long-term suppression of a feeding response by inhibiting those NPY-containing neurons. So I'm going to show you this again in a slightly different form on this next slide. And for the time being, we're really going to concentrate just on the neurons, and we're not going to worry about these major influences on hunger right now. The only one that I'm going to concentrate on is glucose. I'm going to come back to each of these in turn on the next slide. So that's where I'm going to concentrate those on, but you should be able to come back and figure all of that out. So here is the hypothalamus. This whole structure up here is known as the hypothalamus. Down here we have the third ventricle, and this is how we organize ourselves in looking at the hypothalamus. So this third ventricle you'll see over and over. You'll see the suprachiasmatic nucleus in relation to this third ventricle as well. And this is that arcuate nucleus that we talked about in that previous slide. This arcuate nucleus has two groups of neurons. Here is one group of neurons, and here is another group of neurons in the arcuate nucleus. This group of neurons, what it has as a neurotransmitter is neuropeptide Y. When it gets activated, this will signal up, release neuropeptide Y, and it will then activate this group of neurons known as the paraventricular neurons up here. And when these neurons get turned on by neuropeptide Y release, these will then cause you to start feeding. It will initiate a hunger network or a feeding network that will then activate those pre-programmed responses in the periaqueductal gray that allow you to start eating. So this is that one group of neurons that I mentioned to you before. So what if you're not hungry? What if you have a full stomach? What if you have lots and lots of leptin being released, and what if you're activating the other pathway? The other pathway is this pathway that's outlined for you here on this side. And on this side, it's this melanocortin containing group of neurons, also found in the arcuate nucleus. And what it does is it will signal to another separate group of neurons as part of the satiation. I feel full, I feel like I don't need to eat group of neurons, and it does so by releasing the neurotransmitter melanocortin. And I told you this other group helps to modulate and turn up or turn down the activity of these other groups of neurons. And it does so by sensing glucose levels in the blood that's flowing by it. And as the blood glucose levels change, it will change the firing of neurons that are found in this region known as the ventral median nucleus. So this will then inactivate, for example, the NPY containing neurons if you don't need to be able, if you don't need to feed, if you have lots of blood glucose. And as I mentioned earlier, I'm going to talk to you about some of those other groups of modulators in the next slide. Should you expect a couple of questions related to the circuitry of NPY versus melanocortin versus arcuate nucleus? Yes, you should. I'll make it as straightforward as possible as long as you know this diagram and you know this particular slide that accompanies this diagram, you'll get perfect on that question. But I do realize that there are a lot of nuclei that we've just talked about here today. So there are a number of different hormones that actually can go from your stomach and from your pancreas and from your gut through the bloodstream directly up into the brain and influence the brain. And especially at the level of the hypothalamus that we've just been looking at. One of these hormones is known as ghrelin, and ghrelin is released from the anterior portion of the stomach, from the fundus of the stomach every time it starts growling. So when you hear that growling noise, like when you're really, really hungry, right, and you hear that growling noise, that's a good thing because it's signaling and the release of ghrelin. And ghrelin is going to work its way up into your brain so that you feel hungry and you'll start initiating feeding behaviors. So ghrelin is a very powerful positive modulator. So this is why it's in green and a big plus sign. And it is an orexigenic hormone. It's a hormone that wants you or initiates a feeding response in the brain. So orexigenin is a big word that we'll come back to a little bit later, but it is a feeding hormone, in other words. Have any of you heard of PYY? Can you guess what PYY stands for? Not a very adventurous group. Any ideas? Sorry? 3 amino acid peptides. So it is a small peptide, absolutely, and it is peptide YY or peptide tyrosine. And this small peptide, this small signaling peptide that involves tyrosine is released from the intestine in response to food being in the intestine, as well as insulin from the pancreas. And what it does is it travels to the bloodstream and it will inhibit the feeding behavior at the level of the arcuate nucleus. Other signaling peptides, like other signaling molecules like cholecystokinin or CCK, as well as OEA, and here's the long form name of OEA, which is a fatty acid derivative that gets secreted by the small intestine or the duodenum, will activate the vagus nerve, and through the vagus nerve, it will signal through the nucleus tractus solitarius to inhibit feeding behavior through a direct neuronal connection up into the brain. And this is another slide that I'd like you to spend some time on, a pretty simple slide in terms of the regulation and modulation of feeding behavior. What things turn on feeding behavior, what things turn off feeding behavior. And you'll either see it on the term test or you'll see it on the final exam. Do you have any questions about that before I go on? So I told you earlier we have a number of autonomic brain centers, and these autonomic brain centers can actually turn on different programs related to feeding, related to the limbic system, etc. And the amygdala is one part of the limbic system. It's actually considered part of the basal ganglia, and this modulates and changes autonomic types of behavior, including things like eating. So it receives input from the limbic cortex, things that will eventually lead to the autonomic brainstem activation. And the amygdala is really important because it sends output. It sends axons, which are generally inhibitory to turn off the periaqueductal gray, those premotor programmed feeding behaviors, etc., as well as the hypothalamus. So when you want to turn off the hypothalamus, you turn on the amygdala. When you want to turn off the periaqueductal gray, you turn on the amygdala because it's usually inhibitory. Many of you will know, because it's part of the limbic system, that it is involved in fear responses, and these fear responses can actually result in a number of uncontrolled autonomic behaviors. So some of these autonomic behaviors that again involve some emotional components involve things like sham rage or hypersexuality, etc. Generally speaking, for those of you who have either taken first year psychology or other physiology courses, what is the amygdala most tightly associated with or closely associated with? Yes? So a fear response. So imagine you have wiped out the amygdala, which is involved in a fear response, and you no longer have fear. What does that look like? What if you weren't afraid? What would you do? Sorry? You would take PSL300. Okay, good. Nice. So this actually occurs much more often than you would like to think. You don't have to actually physically damage it. Some individuals, as you get older, your amygdala actually, because of the blood flow to the amygdala, it undergoes calcification. So in terms of what types of things happen, this happens. If you're no longer afraid of what people think of you, then you start doing all sorts of weird things that relate to autonomic behavior. And again, some of that is hypersexuality, some of it is related to loss of emotional context and fear, etc. And so this is Erica Latour Eiffel. She had married the Eiffel Tower because she really didn't care what anyone thought of what she was doing with the Eiffel Tower. And yes, she did have sexual relations with the Eiffel Tower, because she didn't really care. She didn't care what society thought of her. And it was well documented that she had lesions to her amygdala that led to this type of abnormal sort of autonomic behavior. And this gets into emotional related memory, again, relying on the amygdala. We won't really worry about this particular section. I know that Dr. Key covered a little bit of this with you. The amygdala is not where memory is believed to be stored. Do you guys know where memory is believed to be stored? It's actually a little bit of a trick question. Any ideas? Any psychology students? Other physiology classes? Yes. It's not stored in the hippocampus. That's a very common sort of way of thinking about it. But the hippocampus is really a USB drive. If you want to store things on the hard drive is really the cortex. And that's where memories are stored for very long periods of time. It's really out here in the cerebral cortex. So memory, even emotionally related memory, is really not part of the amygdala. It's distributed across the limbic cortex, that we'll talk about in the next slide, as well as mediated through the entire cerebral cortex that you see here. So it tells you, for example, that if something is bad for you, if something is dangerous, you shouldn't engage in that type of fearful behavior, and it might be a good time to not do or engage in that type of behavior. Again, this gets into the limbic cortex, and I believe, if I'm not mistaken, Dr. Key covered most of this with you as well, right? The hippocampus and other regions of the brain. Okay, I might be mistaken. And this limbic cortex, again, it's really an older part of the brain. It's, even though it's part of this cerebral cortex, it's an area of the brain that very heavily influences the type of behaviors, even autonomic behaviors, that you engage in. Remember at the very top, the cerebral cortex influencing sort of the autonomic behaviors that get influenced by the hypothalamus. It's because of the limbic cortex that we are able to control and modify different types of autonomic behaviors. And I had made the assumption incorrectly or erroneously that you'd already covered that with Dr. Key. I won't go through that, nor will I ask you to know the details of this slide, because usually he covers that in the first few lectures that he goes through. Nor do you have to really worry about this slide, although you've seen different aspects of this before. So we have a choice. So I have about 8 more slides to go through. Would it be okay if we worked our way through the break and we just left early at, say, about 7:20 today? Yeah, would that be okay? Great. Okay, so I'm going to start off a little bit differently. Who do you know in these slides? Do you know any of these individuals? No? So you don't know Michael Cork? No? No idea who Michael Cork is, nor do you care about Michael Cork? How about John Allen Jordan? Do you know John Allen Jordan by any chance? So these two individuals, how about this? Do you know this acronym, FFI? You guys are looking at me like, no, I've never heard of FFI. Have any of you heard of FFI at all? Have any of you heard of prion diseases? Yes, you heard of prion diseases. Um, so I started off today talking about autonomic behaviors, and we started looking at feeding behavior, we started looking at homeostasis, we started briefly talking about how the hypothalamus controls all of these things, and we talked very briefly about different types of nuclei that are found in the hypothalamus. I mentioned to you in one of those slides that circadian rhythm is controlled at the level of the hypothalamus. And I mentioned in passing that what would happen to you if you stop sleeping? This is what happens to you if you stop sleeping. So in 1981, the first incidence that was recorded, at least in modern history, was recorded in Italy. And this individual that was recorded in Italy, there is no patient name associated with him, he couldn't sleep. And because he couldn't sleep, he wasn't able to do very well, and because he wasn't able to do very well, he eventually died. No one really made a lot of his particular death. A neurologist at the time sort of classified that as an abnormal neurological function. Michael Cork is the first individual in the United States to actually have been documented with what is now known as FFI, or fatal familial insomnia. You cannot sleep. You cannot sleep ever, and it happens as a neurodegenerative condition in your hypothalamus. And you'll understand why by the end of today's lecture. And you don't have to take down a single note because you don't need to worry about FFI at all. It's just to give you an idea of what sleep is all about, because that's what we will be focusing on. Michael Cork is diagnosed as having, because he can't sleep, people thought he was a drug addict. You can't go to sleep, you must be on drugs. You're not on drugs, you must have multiple sclerosis. And this is in the early 1990s, by the way. So it's not like a long time ago. Modern medicine was trying to diagnose this individual who would never encountered this type of thing, where this person simply could not fall asleep. So what would you do as a doctor? Your patient has shown up on your doorstep. You have a neurological condition, you think, where you can't fall asleep. So what do you do to help him fall asleep? You give him prescriptions of what? Sleeping pills. Okay, sleeping pills. He takes a whole bunch of these benzodiazepines and other things that would normally cause most of you to fall asleep. Does nothing for him. He still cannot sleep no matter how many sleeping pills he takes. He continues to lose weight. He continues to have dementia. He's only 40 years old. Because he can't sleep, by the time he dies 1 year later, he has full-blown dementia at the age of 40. It looks like he has Alzheimer's after 1 year. And so this is fatal familial insomnia because you can't sleep. The same thing happened to John Jordan. And again, it's an inherited disorder. John Jordan lasted 6 months longer. So he didn't die after 1 year, he died after 18 months. He actually also took sleeping pills, no effect whatsoever. He engaged in mindfulness meditation, tried relaxing techniques, none of those worked. The doctors actually tried to induce a medical coma in him, but even in a medical coma where his eyes were closed, he was still not fully asleep, and so he died 18 months later. So this is still, to this day, something that we don't encounter very much. There are only about 6 families that have been identified in various parts of the world that have the autosomal dominant form of fatal familial insomnia, but all of them will eventually, by the time they reach somewhere between 40 to 50 years of age, all of them will die because of a lack of sleep. And there's no way for anyone to actually fix the situation that they have. So why is sleep important, and what are the different types of things that initiate sleep, and how could we understand the importance of what regulates sleep in terms of understanding things like fatal familial insomnia? So we know that circadian rhythms are really important to help modulate things like body temperature. And circadian rhythms, again, as I'll show you, are really the day-night cycles that you encounter every single day. These are different than the ultradian short-term things that might occur within like a few hours. So the day-night patterns that get established are really important in terms of how you synthesize and release hormones, how your body temperature goes up and down during the day, how autonomic behaviors, which we just talked about, including feeding. Most of you are likely going to eat as soon as you wake up in the morning. Most of you are going to sleep, most of you are going to eat when it's about 6 or 7 o'clock at night because you get hungry. And it's because of a circadian rhythm that is influencing your autonomic behavior. Most of you are going to feel, as young individuals, are going to feel pretty grumpy in the morning, and you're going to feel pretty mellow at around 4 or 5 o'clock in the afternoon. Your level of alertness is also locked into a circadian rhythm, but most importantly, that sleep-wake cycle that I mentioned to you is closely associated with this circadian modulation. This is just for your own information. This is sort of a typical sort of pattern that you would have if you were falling asleep. So again, I'm not sure why they keep putting this up here. As an older individual, I can guarantee you I don't go to sleep at midnight. If I'm awake past about 10 o'clock, it's usually because I have insomnia, not because I'm intending to stay awake. But following some of you on social media, it looks like most of you are falling asleep somewhere between 1 and 2 o'clock in the morning. So it might be somewhat appropriate. So you are in this awake pattern, and you'll go up and down through these different patterns of sleep throughout the course of an evening. So you'll go down into what's known as stage 1 slow wave sleep, and then you'll get into these non-REM patterns, and you'll go up and down through these. Eventually, at some points in time, you'll enter into this REM sleep that most of you will know is that rapid eye movement type of sleep, where maybe dreaming occurs, maybe it doesn't. Again, a lot of controversy about it, but it seems that when your brain needs to reset itself, remember sympathetic, parasympathetic, resetting what's been lost, it's really these periods that seem to be important in allowing your brain to recover or to help consolidate memories. And this is one of the importances of things like sleep. So this circadian rhythm that allows you to fall into this period of sleep and then to wake up, stay awake for 12 hours before you get back into this sort of 6-hour cycle again. These circadian rhythms seem to be entrained within this 24-hour period. Okay, so you're awake, and then you have this 6-hour, 8-hour block of sleep where you go up and down into REM. And again, you don't have to worry about this previous slide. And how does your brain know? Because it's so important. How does your brain know when to fall asleep, when to stay awake? How does your brain know that it's following this circadian rhythm? So we think that this gets entrained into this 24-hour cycle from the retinal input from those ganglion cells that contain melanopsin. And we talked about that at the very beginning of today's lecture. Those retinal ganglion cells that contain melanopsin that look at luminance levels, how bright it is, how dark it is, and that will send projections back into the hypothalamus. They send projections into the hypothalamus at a group of neurons in the hypothalamus known as the suprachiasmatic nucleus. They will also send retinal projections, as I mentioned to you before, into this pretectal area. Do you remember the pretectal area where we started organizing the pupillary reflex? And this pretectal area that, again, as part of the superior colliculus, sometimes it sends sympathetic efferents, sometimes it sends it to the superior cervical ganglion, and sometimes it's involved in the pupillary reflex, but sometimes some of those signals are also sent to the pineal body. And this pineal body is important in releasing hormones like the melatonin that I'll talk to you about in a few moments. So again, this is again to orient you. This is that third ventricle, and remember the arcuate nucleus would be sitting outside the third ventricle, and you can see at the bottom here we have these suprachiasmatic nuclei, and it's very close to where the optic inputs come in. Remember, the optic chiasm is where the left and right eye send their axonal fibers back, and then they bifurcate. And this is that optic chiasm, and here is right where the SCN is sitting, the suprachiasmatic nucleus. So it's right on top of the optic chiasm, getting input from those retinal ganglion cells that contain melanopsin, and it's found on both sides. It's a paired structure on either side of the the third ventricle, and it's sitting right on top of this optic chiasm. This group of neurons is the one that controls your circadian rhythms and whether or not you fall asleep or whether you decide to stay awake. So this 24-hour oscillator that involves cells within the suprachiasmatic nucleus is really the master regulator. So it might surprise you that it's more likely that you will die of a heart attack based on a circadian rhythm. It's more likely that you will suffer if you have bleeding, for example, or you have a cut, that it follows a circadian rhythm. Most of our behaviors follow a circadian rhythm that is entrained at the level of genes that are found in this group of suprachiasmatic neurons. There is a feedback loop that unfortunately I can't talk to you about today, but this group of neurons in the suprachiasmatic nucleus is this master circadian clock. It regulates all of those features that I talked to you about, including the most important one that I want you to know related to sleep-wake cycles. So this group of neurons is modulated again by retinal input. So those retinal ganglion cells that contain melanopsin, that's the retinal input. It is also vulnerable or modulated by pineal input, and the pineal input is going to be coming up in the next few slides. And these are the seasonal variations in the level of melatonin that get released. So in where you have lots and lots of sunlight, the level of melatonin is very different than when you have very little sunlight. And this pineal input will modulate this 24-hour clock as well. Another thing that modulates the suprachiasmatic nucleus is this reticular activating system. And this releases serotonin, and serotonin, as I mentioned to you earlier, will also have effects on your awake, your alertness, and other factors that I mentioned at the outset. So here's that pineal gland that you see here. So right on top of the third ventricle, it's surrounded by the cerebral spinal fluid and various blood vessels. And the importance of the pineal body is that it can secrete melatonin directly into the hypothalamic region, into the CSF, as well as into the bloodstream. This pineal body is really important because it gets activated in the darkness. And again, this is an important slide. You guys all have this slide, correct? Yes? So this is an important slide for you to come back to review, that this pineal body that releases melatonin is activated by the sympathetic reflex system in the darkness, and this gets back to the pupillary light reflex. It secretes melatonin from the pineal body, and it's not a neurotransmitter, it's more better to call it a neurohormone. The release of things like melatonin is blocked by even small amounts of light. So blue-green light tends to block the release of melatonin from the pineal body, and blue-green light is exactly what you're staring at the screen. It's what you stare at before you go to sleep when you're looking at your tablet or looking at your phone or you're looking at a TV screen. That's sort of the blue-green light. Surprisingly, red light has no effect on the secretion of melatonin from the pineal body. Why again is that important? Because melatonin is what is believed to modulate these circadian rhythms involved in sleep induction. So the maximal secretion is already when you're supposed to be deep in sleep. Remember that diagram that I showed you? Again, if you're young, you start sleeping at about 12 o'clock, and by 2 o'clock, you actually have the maximal levels of this melatonin that is supposed to help you stay asleep. But again, if you're staying up and you're looking at a blue-green screen, you're not being able to cause the formation nor the release of melatonin, you're not going to get that level of sleep that you normally should have. The other thing that I would like you to know, and forget the numbers like 2 a.m., but the other thing that I'd like you to make sure that you know is because it is sensitive to light-dark cycles, counterintuitively, it's actually secreted at higher levels in the summer than it is in the winter. Where you think that it makes more sense to be able to sleep longer, especially in light of the pupillary reflex, melatonin actually is secreted at higher levels during the summer than it is in the winter, but it also undergoes seasonal variation. So this is probably the most important slide that I wanted to talk to you about. And this slide is one, as well as the final slide, one that I want you to spend some time on prior to the upcoming term test, as well as the final exam. So these sleep-wake cycles involve various groups of neurons that are found in the hypothalamus. So this as a whole is the hypothalamus. Here's the pituitary gland down here. Here's that optic chiasm, and sitting right on top of the optic chiasm is the suprachiasmatic nucleus. This is the master regulator. Some neurohormones like melatonin will influence how much the suprachiasmatic nucleus fires or how it gets inhibited, but this is really what starts this whole sleep-wake cycle. So you'll see, importantly, the suprachiasmatic nucleus within the hypothalamus actually sends axons or projections to two different areas. So one, it will send groups of axons to this the VLPO, and we'll just keep it as a VLPO. And another one, it will send a projection into the TMN group of neurons, the tuberomammillary nucleus, as I mentioned to you before. So it makes sense that just like we looked at before in terms of the rostral caudal and the two different central pattern generators in your spine, when we have two opposing actions, that they will have some reciprocal inhibition. So when the VLPO neurons get activated by this activity of the suprachiasmatic nucleus neurons, you will start to fall asleep, and it inhibits the tuberomammillary nuclear group of neurons that keep you awake. Okay, so this, when it gets activated, VLPO induces sleep behavior in you. And when you need to wake up, for example, there are other factors that come into play. One is that in the suprachiasmatic nucleus, one of the things that happens is within another area of the hypothalamus, you have this group of orexigenic neurons. I told you earlier what orexigenic means. Do you guys remember what it meant? Sorry? It induces feeding behavior, it's hunger inducing. Originally, this group of neurons was thought to induce feeding behavior because, again, in the hypothalamus, many things are found to induce feeding. But it turns out that this group of neurons that gets activated by the suprachiasmatic nucleus, actually this hypocretin or orexin-containing group of neurons that you see here in gray, when it gets turned on, it causes the release of orexin that will then activate this group of TMN neurons. So by doing so, this will then activate and this group of neurons, which turns off the sleep group of neurons. So they're kind of balanced out. They are antagonistic to each other. The suprachiasmatic nucleus can control either group. And somewhere in this mess, where you're looking at either the activation of a sleep pathway like in the VLPO, or you're inhibiting the wakefulness pathway, somewhere in FFI, different prions are believed to cause the degeneration of the different groups of axons that regulate this pathway. And it's not really clear whether it's the VLPO only neurons or it's this whole group that gets lost in things like fatal familial insomnia. Again, the most important things for you to remember are the VLPO are induced in sleep responses. This orexigenic group is then targeted to cause wakefulness types of behavior. And then when wakefulness is occurring, the TMN groups of neurons have to be fully activated and fully awake. Do you remember, and again, this is the way I want you to think about this. The TMN group of neurons, what was the neurotransmitter that it released? And that's how you should study for this slide for your final exam and for your term test. Do you remember it all? Does histamine ring a bell for any of you? So again, histamine is released from the TMN group of neurons, and we talked about antihistamines and feeling drowsy, etc. So this sleep switch in that VLPO or ventrolateral preoptic nucleus is really important, and this activation of VLPO by that circadian clock through the suprachiasmatic nucleus, which could get turned on by things like melatonin. So melatonin turns on the suprachiasmatic nucleus, which then turns on and activates the VLPO, allows you to get to sleep. Other things that turn on the suprachiasmatic nucleus is sleep debt. So when you are staying awake, one of the things that happens is your brain undergoes what's known as sleep debt. Have you guys all experienced sleep debt? Of course, sure, you're all U of T students. Who wouldn't have experienced sleep debt? Sleep debt is really simply this. What happens to you if you stay awake for 48 hours straight and then decide to go to sleep? Do you go on a normal cycle of sleep? You sleep a lot longer than you normally would. And part of the reason why you do that is because you are in sleep debt. During that time that you've been staying awake, you have used up the very small stores of glycogen that exist in your brain. Those stores have dropped. And in addition, adenosine, which is a metabolic byproduct, has built up in your brain. Adenosine, as I mentioned to you before, is inhibited by things like caffeine. But all of these things are things that will be detrimental to you to be staying awake. The waking switch is found in the TMN, or that tuberomammillary nucleus, and involves that histamine that I talked to you about before. It activates, or it's excitatory to brainstem activation, and it in turn can also be modulated by that group of neurons here, that orexin hypocretin containing group of neurons. And both of these centers, when one is on, the other one has to be inactive. Do you guys want a preview of your term test? Yeah? Okay. So there is instead of not being able to sleep, where you have fatal familial insomnia, do you guys know of any other sleeping disorders where yes? So I don't know if you've ever heard of cataplexy or narcolepsy. In cataplexy or narcolepsy, you fall asleep randomly. Like you might want to be staying awake, you might be crossing the road, and then suddenly you'll fall asleep. It's not something that you would normally want to do. So I want you to think as a class, because you'll likely see it, what group of neurons do you think are going to be more active in places like, in conditions like narcolepsy, which cause you to fall asleep at inappropriate periods of time outside of the circadian rhythm? And you will see a question like that on your upcoming term test, and think about these groups of neurons that I just mentioned to you. So some final thoughts for your test. So spend some time reviewing the general features of the autonomic nervous system, the location, the myelination patterns. Think about the general features of the sympathetic, parasympathetic, which ones have longer preganglionic, which ones have longer postganglionic, if they're myelinated, etc. This is all on YouTube, by the way. Feel free to take pictures. I think the YouTube link is still running. Yes, I think. I'm hoping it still is. I'm pretty sure it still is. Think about all of those different neurotransmitter, diffuse modulatory systems, the origins, the groups of nuclei that are associated with histamine, TMN, histamine, raphe nuclei, for example, serotonin, and think about other neurotransmitter systems that we looked at. Think back to the arcuate nucleus, nuclei related to feeding, related to blood pressure regulation, and sleep-wake cycles, the suprachiasmatic nucleus sending projections to the next group. I never had the opportunity to take a look at your midterm before, so I got an opportunity because someone dropped by. The next term test and the final exam, I'm just going to make it really straightforward, more like the questions that you're used to. I was told to make them somewhat challenging, but I looked at it and I didn't find all of the questions challenging. Only my questions were kind of like that type. So I'll make the other ones much more straightforward, and I think it'll be something that you're a little bit more accustomed to. There'll still be one or two sort of thought questions or integrative ones, but overall it should be something that you're more comfortable with. And I've been asked to submit 21 questions for this section, the last 2 lectures plus today's lecture, so 7 questions from each lecture, in other words, and then 14 overall for the final exam. And for both the final exam as well as for the upcoming term test, I'm going to make them a lot more straightforward, where you just have to know the answer without thinking too much about it. These are the types of questions that I asked previously, and in theory, you should actually, based on what you know today, from today's lecture, you should be able to answer this. And this is the type of question that I'd like you to be familiar with. Again, it'll be even more straightforward than this, but you should be able to answer this based on what we just talked about here today. So I'm hoping all of you will be picking the raphe nucleus. And again, if some of you are interested in getting previous test questions to see what types of questions I asked previously, these will be included in that test question package. And again, this is sort of the type of question that I would like you to make sure that you're familiar with. And this is an example of a straightforward type of question that you might see related to stimulating to increase the rate of flexion extension in the step cycles of walking? Straightforward question. Do you know the answer? Any guesses? So do you remember what the cingulate motor area is used for? No? Is it used to increase the rate? It's not. The rubrospinal red nucleus, remember synergies, fine synergies relate to distal digits and limbs, not step cycle. So you really have a choice between looking at the lumbar spinal cord or looking at the MLR. Which do you think? 50-50 chance. A or D? A or D? So the correct answer is the mesencephalic locomotory region, because if you remember from that lecture, it's taken directly from the notes. It's a straightforward question. As long as you've studied it, it's really an increase in the frequency from the MLR causes an increase in the step cycle of walking. And that's an example of a straightforward type of question that you should expect to see on your upcoming term test. And please do email me if you want an exam package. Feel free to share it on your Facebook study group or to use it in your own sort of private studying.